Okay, so this is part 2 of the chapter 13 lecture. What viruses do once they enter a cell is they replicate. They make new virus particles, and the speed at which they're able to do that determines how quickly the infection can spread. So one virus particle enters a cell. In this slide, we're showing, on this slide, I'm showing some of the language that is associated with bacteriophage. And bacteriophage would be, let me get a pen. Bacteriophage is spelled like this. Bacteriophage. Boy, that's terrible. Bacteriophage. And we usually call them phage for short. But the language that's used here, even though it's specific to bacteriophage, the concepts cross over into how animal viruses, for example, infect and how they replicate. So for bacteriophage, we typically talk about the lytic replication life cycle and a version of it called the lysogenic replication cycle. So the lytic cycle, which I'll show you on the next slide, is a cycle in which the virus enters the host cell, and the chromosome ultimately is the thing that gets into the cell. The chromosome contains genes, and the cell's enzymes and the cell's ribosomes transcribe and translate those genes into proteins and enzymes needed to make more virus. So the end of that results in, or the end of that cycle is when the host cell dies because it bursts and it releases all these new virus particles that have been replicated, that have been made. The lysogenic cycle is really a modification of the lytic cycle that begins with the virus chromosome entering the cell, incorporating onto the cell's chromosome, and then when the cell divides and grows, it copies that virus as part of its own chromosome, and the virus is dormant. Sometimes we call that the provirus. And then once it, for whatever reason, reactivates, then the cell then goes into the lytic cycle and ends with death of the cell. So you have some types of animal viruses that function pretty much like this. Either they enter a cell and replicate and cause the host cell to die when they are released from that cell, or they have a period of dormancy where they can integrate into the host cell chromosome and stay dormant for a period of time. So these concepts are crossover into animal viruses as well. All right, so here's a picture of the phage, the bacteriophage, and you can see the title of the slide, the lytic replication cycle of bacteriophage. And the first picture at the top, sort of on the left, shows you a typical phage. This is the one that has that complex structure with the polyhedral head and the helical tail, and then the little spider legs. They're called tail fibers. But not all phage look exactly like this, but whatever the shape of the phage, there is a chromosome of some type, and the phage lands on the surface of the cell, in this case the bacterial cell, probably an E. coli, and the chromosome is delivered into the cell, just the chromosome. So the capsid portion of the phage does not enter the cell. The protein portion of the phage does not enter the cell, but the chromosome does. And the chromosome has genes on it, and the genes are transcribed and translated to make phage proteins and phage enzymes to build new phage. And so you can see on step 3 and step 4 how the phage is using the host cell's machinery to build new phage. And the phage are assembled, and then in this picture there's 5 phage, but think about it being like 500, not just 5. And so the cell, the very last step of this is that the cell bursts, lyses, and that releases all of these new phage. And each one, they're showing step number 5, they're highlighting one of those phage from step 5, then goes to a new cell. So each of the 500 phage that are released, each find a new cell to infect. So very quickly this infection will spread exponentially through a bacterial culture. So in phage, when we think about phage replication, we typically think about what we call burst size, because when one phage infects a cell, then it can result in the release of hundreds of new phage from that cell. And so the number of viral particles that would be released from an infected cell during one cycle of the life cycle is called the burst size. And it depends on the virus, but you want to think of burst size being, 100 or more, sometimes 1,000 per life cycle. So one virus can become 100 or 1,000 within less than an hour, typically. And it depends. That amount of time depends on what kind of virus it is. But so in this picture, you have the big oval is an E. coli, or a bacterial cell, a bacillus shape bacterial cell. It does say E. coli. Yeah, it is an E. coli. And then it's being infected by a whole bunch of viruses, all of these phage viruses. But what you can see on the inside of the cell are all the new phage that are being built. So you get a sense of this, the numbers, what would be inside the cell. And this is a transmission micrograph, so you're only getting a 2D slice of the image. But in 3D, you can imagine how many virus particles that would be. So it's a lot. So this is how virus grow. We looked at a concept of how bacteria grow exponentially, logarithmically, and that's because each cell, when it divides, becomes 2, and then 2 divide, become 4, and 4 divide to become 8, and so on. And that is logarithmic, but it's not, the growth of a bacterial culture is not as logarithmic, or is not as steep of a slope as a virus, because like we said, the virus, when it replicates, it doesn't just make 2 virus, it makes 100 or it makes 1,000. So the burst size makes this growth rate enormous compared to a bacterial infection. So in this picture on the far left, you see, let's say one virus particle enters one cell. And so you have, at time 0, you have the entry of the virus into the cell. And it takes, maybe 20, 25 minutes for the replication whole process to happen. But close to 30 minutes later, you have the burst of that cell, and you now have hundreds or thousands of virus now. So every 30 minutes, you multiply it by the burst size. So one cell becomes, let's say the burst size is 300. So one cell in 30 minutes becomes 300. Each of those 300 cells in another 30 minutes produces another 300, so 300 times 300 after an hour. So you can see quickly how you would multiply by that burst size for every generation time that passes, and the number of virus becomes enormous. Now this is the lysogenic cycle. Again, that term would only be used for phage. But what you can see here is you've got on the sort of the top left the typical lytic cycle. So number 1 is attachment. The phage attaches to the outside. Number 2 is entry. And then in the lysogenic cycle, you take a kind of a diverted path towards the right side of the screen where the phage chromosome integrates onto the host cell chromosome. So here they're showing it as sort of a greenish blue, becomes part of the purple host cell chromosome, and it gets copied. The cell doesn't even know it's been infected, and it copies it every time the cell divides, but at some point that virus becomes reactivated. And that is step 5 here, where the chromosome then starts taking over the cell. The cell starts making new virus particles, and it ends in the lysis of the cell, just like the basic lytic cycle did before. So animal viruses aren't hugely different in concept. Ultimately, you have the virus has to enter the host cell. There's more ways that animal viruses get into host cells than the ways that phage get into their host cells, because viruses, they don't have the tail fibers. They're typically going to either fuse with the cell membrane of the host cell, or they're going to be taken in by endocytosis into a vesicle, and then that is followed by the release of the chromosome from the virus, or they're just going to cross over the membrane somehow. But the point is, somehow the animal virus gets into the host cell. And one way that we try to treat or try to slow the progression of a viral infection is we try to find drugs that will block the ability of the virus to get into the cells. But I think you can tell that even in this picture, there's only one virus attacking this cell. In real life, you probably have thousands of viruses attacking any single cell. So it's really hard to find drugs that are effective. They have to be almost 100% effective against this entry. So it's been very difficult to find drugs that work, virtually impossible to find drugs that work at that level of percentage, because you're really needing 100% prevention of entry into the cell. Because once the virus is in the cell, then it starts to replicate thousands of copies of itself. Now, a lot of viruses have their chromosome is made of RNA. And the RNA can be, if you just look at this picture from a global view, just kind of sit back and look at it a little bit. Don't get too hung up in every little detail. But the virus can have either a single-stranded RNA chromosome or a double-stranded. So SS means single-stranded, DS means double-stranded. If there is a single-stranded RNA chromosome, then either the single-stranded RNA behaves just like an mRNA, and the proteins needed to make new viruses are translated directly from that single-stranded RNA. Or if it is a single-stranded RNA, but it's complementary to the one that could be the mRNA, then it can be copied into the complementary version and used directly. Or if it's double-stranded RNA, it has both. So the concept here is they're showing in red what we call the plus strand, the strand that can be used directly as mRNA. They're showing the minus strand as the one that is complementary to the plus strand. So if you have the plus strand, then you can produce proteins directly from that strand using translation. If you have the minus strand as the chromosome, then you just need an enzyme to make the complementary plus strand, and that strand is then used as the mRNA. And if you have double-stranded RNA, you have both. So because RNA can be copied into complementary copies, just like DNA is copied into complementary copies, because that's possible, then these RNAs can be copied and can be used in the cell as the mRNA for making virus proteins. There's one special virus that I want to mention, and that's not, well, a special group. It's called the retroviruses. They are RNA viruses, but what they do is they copy their RNA into DNA. And that is the reverse of the way a normal cell makes a copy. So normally cells copy, they either copy DNA into DNA, we call that DNA replication, or they copy their DNA into RNA, and we call that transcription. But cells don't copy their RNA into DNA, but viruses do. There are some viruses that can do that, and that's called reverse transcription. And what they do is they copy their RNA chromosome and make it into DNA, and then they insert that DNA into the cell's nucleus, just like that lysogenic cycle of the phage. In a similar way, these animal viruses can become dormant. Their chromosome can be inserted into the host cell's chromosome, and they can become dormant. And so retroviruses have that ability. And ultimately, whatever type of chromosome a virus has, it makes the enzymes and proteins needed to build new viruses. And then you have two kinds of outcomes. If it's a naked virus, if it's a virus that does not have an envelope, then usually a lot of times that cell is going to burst or lyse to release all of those naked viruses. So that would be similar to the lytic infection cycle of the bacteriophage. If you have an enveloped virus, the animal cell can stay alive, and it can just allow viruses to bud off of its surface indefinitely. And that's even a little bit more problematic because the cell, the infected cell, stays alive, and it just indefinitely releases new viruses. And we call that kind of infection a persistent infection. Instead of the cell just in one big burst releasing new viruses, the cell stays alive, and it just kind of buds off a constant dribble of new virus particles. And that's the way HIV works. So when an enveloped virus is being released, you may recall in part 1 of this lecture, I mentioned that the envelope is provided by the host cell, but maybe it's taken from the host cell. And so here's the picture of the virus particle as it is budding from the host cell. And this is just one virus particle going through this process, but the virus pushes against the plasma membrane, and in a way that's basically opposite of making a vesicle, it pushes out and it buds off of, and basically you get a vesicle that pops off of that cell. And so then by step 5, you have the separate virus particle that has been released from the infected cell. And so when you have a persistent infection, do you remember the graph I showed you earlier when there was a big burst of virus from lysis of the host cell? There was a very steep slope of the curve. Well, when you have a persistent infection, you don't have such a steep slope, but it keeps going up, up, up over a much longer period of time because that infected cell continues to release viruses and it doesn't die. So you don't have just such an abrupt increase in the number of virus particles, but it's more, it goes over a longer period of time. And like I said, just like bacteriophage can have a lysogenic cycle, a period of dormancy, in animal viruses we call that latency. So you want to be careful about the language here. For bacteriophage, we use the language of the lytic cycle, L-Y-T-I-C, and lysogenic, L-Y-S-O-G-E-N-I-C. So lytic and lysogenic, those refer to ways that bacteriophage interact with their bacterial host cells. With animal virus, we use another L word, but it's latency. So, but it's similar to the lysogenic concept in that a latent virus means it's dormant. It's entered the host cell, the chromosome has somehow integrated onto the host cell chromosome, and now it's existing in a dormant phase, so it can reactivate at any time. So that's the end of my second section of our lecture, and I will pick up with this next part of cancer on lecture segment 3.